Hello there and welcome to Expert Insights Volume 7. I'm your host Mark Jeffries, and in today's episode we are diving into the world of biopharma processing, how it's evolving, what challenges are shaping it, and how innovative technologies are helping to create a safer, more efficient future for both patients and manufacturers. Today's episode is all about bringing biopharma processing to life and enabling industry solutions that lead to a better tomorrow. I'm joined by two great experts, Graeme and Angela, so let's have them introduce themselves.
Thank you, Mark. Great to be here. I'm Angela Mattis, the Market Development Manager for Biopharma within the Engineered Materials Group. And in my role, I work closely together with our sales and engineering teams to find process solutions for the growing requirements and needs of our customers. And what we have seen in the last few years is a shift in how biopharmaceutical companies approach manufacturing. So speed, flexibility, and contamination control have become more critical than ever. And that's exactly where single-use technologies also come into play. And I'm really looking forward to deep diving into that topic with Graeme and you today.
So I'm Graeme Proctor. I'm the Product Manager for Single-Use Technology here at Proctor Bioscience in the UK. I've got over 30 years in this industry, and primarily my product lines are single-use consumables, single-use tube sets, which can be used as standalone items or incorporated into automated systems.
All right, well, fabulous. Thank you both for your time today. Let's start by discussing the impact of biopharma processing on human health. Graeme, could you share your thoughts on this particular area?
Yeah, certainly, Mark. So biopharma is all about life-saving treatments. Over the years, there have been many technological advances which have helped to save lives, helped to enhance the quality of life. One of which is single-use technology, which myself and Angela are heavily involved with. And that revolves around changing out or replacing the stainless steel systems, the stainless steel pipework, which you would traditionally have in a biopharma drug medicine manufacturing plant. You know, the primary, issue that single-use technology solves is speed to market. So getting, the drug product to market can enhance lives quicker, and one such avenue which demonstrates that is COVID-19. The pandemic, showed that we needed to get vaccines to the world in a very quick manner. So single-use technology enabled that by getting the disposable, ready-to-use tube sets to the drug manufacturers so that they could increase the manufacturing of the vaccine and quickly get that to market to help save lives.
I mean, yeah, indeed, Graeme, time can be critical. And I mean, single-use technologies have enabled biopharmaceutical companies to accelerate not only the delivery, but also the development of critical medicines. So in situations where time is critical, like producing a vaccine or a personalized therapy, quickly scaling up production is crucial. Another benefit is global access. And I mean, in contrast to traditional manufacturing setups, they require significant invest and time to build. And single-use systems can be implemented much faster and at lower costs. So this does not only increase productivity, but also helps bridge the gap between innovation and accessibility.
Outstanding. You just mentioned vaccines and personalized therapy, two very different types of treatments and processes, I imagine. From your perspective, what is the difference between them?
I mean, both fall under the category of biologics. However, when it comes to how they are manufactured, they can be very different. Let's take vaccines or monoclonal antibodies. They are produced in large scale. You see large bioreactors of thousands to 10,000s of liters. You'll see highly standardized systems, and in contrast to personalized therapies like cell and gene therapies or mRNA treatments, you have much smaller batches. So that means one batch could equal one patient. And there's a real need for flexible, sterile solutions that enable close manufacturing, but also minimize product loss in the system and avoid any holdup volume in the system as well. So what I want to add here as well is that novel therapies were originally developed in academic lab settings or in early biotech settings. So, but now the more, let's say, they move towards late stage clinical trials and commercial production, there's a real need for GMP compliant manufacturing solutions.
Yeah, so I mean, one of the things which I like to say about this is that, one phrase, one's about scaling up, the other's about scaling out.
So, listening to that is absolutely fascinating, but are there any specific examples that you can provide that illustrate these benefits actually in action?
Yeah, certainly. I mean, I did briefly mention COVID-19 there. So that's a prime example of the agility which can be put into biopharma. There was a need during the pandemic to get a vaccine to the masses in an incredibly fast way. So previously, or traditionally, vaccine manufacture can take months, it can take years to get that vaccine to the patients in a safe manner. There was lots of fast tracking went on within the industry to get these vaccines out. Otherwise, people would have still been trapped in their own homes, which was the only defense that we had. So single-use technology allowed that to happen because it, as I mentioned earlier, it replaces the stainless steel systems, the stainless steel pipework, which can take a long time to build a facility. Single use is a modular system which can be, brought in. It can be localized into smaller facilities to provide localized manufacture. It can then rapidly help to scale up manufacture and speed to market of medicines. So, to me, it's a prime example of bringing biopharma to life, and certainly getting that vaccine to people. We all remember 5 years ago when the pandemic all started. Has it gone away? No, it's still there, and I guess we all hope that we don't see anything similar, but we have the technology there, and we've learned from that to be able to act quickly should something similar happen in the future.
That's a real nice example. What we also saw during that time, I think, was a shift in mindset. So not only speed, but also flexibility became the priority. So basically, facilities were reconfigured overnight, and manufacturers had to keep up with their production capacity. So not only for one vaccine, but also for multiple platforms. So single-use systems made that possible, and manufacturer could switch their processes without being locked into one fixed process, and that level of agility was essential.
And with that speed and flexibility, I imagine material selection becomes really critical as well.
It does, Mark. So chemical compatibility is a key consideration to comply with GMP guidelines. So testing is essential to ensure the material does not react or influence the drug product. And there are a variety of substances used in a biopharmaceutical process, and they do not all respond the same way. So a proper assessment of chemical compatibility also helps mitigate the risk of extractable and leachables. And USP 1665 and 665 further underscores the importance of material selection. And they also address the risk of extractable and leachables, and they do set out clear expectations of how materials should be evaluated.
Yeah, and I guess it's not just about material breakdown and particulate generation, and items sort of falling apart. It's more about real world scenarios, making sure that those materials are compatible with every process solution that you're going to be putting through. While in practice, or sorry, in theory, you can demonstrate that these materials will stand up to the conditions that they have. There's nothing which beats real world experience and real world practice to get you that data to show that the product that you're putting through is safe and nothing else is getting in there.
Nice. So that's covered chemical compatibility. Are there any physical limitations?
Yeah, there's lots. I guess the first one that comes to mind is pressure. Because these tube sets and the single-use technology is a polymeric or plastic based solution, you don't have the real high pressures you can get to within stainless steel systems. So pressure has to be managed to match the process that's involved and the material which is being used in that system. So take, for example, you could have a tube set which can take up to 4 bar. You could run happily at 4 bar pressure, but if your equipment can't take that 4 bar, such as a chromatography column, which may only be able to go to 3 bar based on the glass walls that it has, you wouldn't want to take it to the extreme of the 4 bar. You would run at the lowest pressure the system could run. And coupled into that, you've also got temperature. So you've got a relationship between the two with viscosities of liquids, with temperatures, with pressure. Within biopharm, you've got extremes of temperature which can be used from low temperature storage, such as -80-degree freezing, all the way up to high temperature sterilization or autoclaving, which can take you up to, say, 130 degrees C. So, biopharm has to take on board all of those extremes of temperature, and any material that you use has to be able to withstand that.
Yes, Graeme, temperature is actually a good point, because you also have to ensure your single-use system's integrity. And you just mentioned -80 degrees C, and I mean, we have often different materials too. Let's take a silicone tubing and a polypropylene cap, for example. And when freezing down to -80 degrees C, you have to ensure that the integrity of your system is still there. So it's not just about selecting any material, it's to ensure it performs well under the just mentioned process conditions. So that means conditions like pH level, temperature, or flow rate need to be considered. So testing is vital. It also needs to ensure the material don't degrade or react adversely, but also ensures, as I just mentioned, the overall integrity is maintained. And these physical properties can also have an influence on extractable neutrables, so they need to be considered as well.
Fascinating insight. Thank you for that. Let's take a moment to discuss the complexity and timeline of the biopharmaceutical process. It's not a quick endeavor, right? It can take months to go from start to finish, with each stage adding significant value to the final product. So, Graeme, I'll turn to you. Can you elaborate on why this timeline is so critical and how single-use technologies can help streamline these processes?
Yeah, certainly. I mean, as you mentioned there in your question, the whole biopharm process can take months, years, to get from the cell culture side all the way through to the final fill into the vial. And traditionally that is the route which has been taken. There's lots of technologies which are currently there, which are, certainly the COVID-19 vaccine showed that there were technologies which could speed up vaccine manufacture. So, that is all, coming on board, and there's more and more advances with that. But there's lots of different stages within the complex process of the biopharm situations, where you've got things like starting at R&D all the way through to clinical trials, to the final fill into the vial, which it then goes to the patient to help enhance their life or to save their life. It's a very meticulous journey. If anything goes wrong at any of those stages, then there's varying degrees of loss for the manufacturer of the drug product and for the patient. So if something goes wrong at the start of the process, so the cell culture side, it generally happens fairly quick. So the amount of time, effort, and materials which have gone into that stage are relatively low with regards to the final cost, or to the manufacturer or the patient. So it can be quickly recovered. If something goes wrong at the end of the process, say 3, 4, 5 months into that process, maybe, then you've got a significant loss, because there's been lots of time, effort, and money gone into that to produce this drug product. And at that point, it's a very valuable item. So if something goes wrong at that stage, not only is it a costly loss for the drug manufacturer, it's also costly because if patients are waiting for that drug, then it's suddenly a batch of that drug fails, and you can't get it to them, then you've got significant impacts on potential lives, potential enhancement within someone's life.
Yeah, as you just mentioned, it's indeed very challenging and can be quite complex. So scaling options need to be considered upfront to ensure also volumes and timings are not restrictive. And also the single-use system design needs to be thought through early in the process, because as more we get towards commercial approval, the harder it gets to implement any changes due to regulatory and economic restrictions.
Yeah, and I guess the whole thing is about compliance and safety and quality. You want to get that drug product to someone in a safe manner, so it does what it should do, but it also is safe and doesn't have any harmful side effects.
All right, lots to think about there. So can we now dive into the applications of bioprocessing? Graeme, again, what are some key applications that single-use technologies support?
Would I say one application is more important than another? No. What I would say is that the whole biopharm process is made up of what I would class as several sub-applications, or many sub-applications. You all the way through from cell culture to final fill. You've got several stages within there which could be repeated multiple times, such as chromatography steps, tangential flow filtration steps, standard filtration, concentration, diafiltration, buffer exchange. All of these come into play. As I mentioned before, it's a very long journey, but lots of different sub-steps in there. Now, I couldn't really classify one as a key standout. You could potentially say that final fill is the ultimate, the end point before it goes into the vial for the patient. But, throughout the whole process, I would probably classify them more of a series of symbiotic steps which all work together to get you that final product.
Nice. Well, you mentioned final fill a couple of times, so let's talk about final fill solutions. How does Parker support this critical stage?
Graeme just mentioned that final fill is the last and most critical, or one of the, or very critical step in a drug manufacturing step. So let me just give you some background on the final fill. So here the drug has its highest value, and I mean there's no room for error, as aseptic processing here is very essential. As Graeme just mentioned, in that step the drug product is filled into its final container, being a vial, syringe, cartridge. And what we focus in that step is on supporting with flexible closed single-use system designs that align with Annex 1 And we also support our customers in upgrading existing filling lines or setting up new filling lines. And the single-use assemblies can be quite complex here at that stage because it does involve many components, many connections. There are large assemblies, and therefore ensuring a system integrity, accurate dosing, sterility, and a fully closed design is very critical. And also overmolding technology can play a key role here that supports just the above mentioned points. And overmolding, just to quickly explain what overmolding is, it's a technology that goes far beyond the manual assembly of a tubing with a hose barb connector and a cable tie. It's a chemical thermal bond that leads to a single seamless product that optimizes fluid flow.
Very interesting. Graeme, let me ask you, what are the challenges then that you see biopharma facing today?
If I had to sum it in one word, I would probably say compliance to regulations. It's a never-changing field with regards to regulations, and especially since sterility is key to make sure you have safe products. The regulations are very, very tight. So I would say probably to me, from myself in my field, in my role, what I'm seeing is there is a challenge in single-use assembly design to make sure that sterility can be offered in the process steps it needs to be in many occasions, because traditionally you had the stainless steel systems which would have been built, the steel pipework would have been fixed, it would have been steamed in place, it would have been ensured sterile, it would have been tested. Now the onus is on the vendor of these single-use items to ensure sterility in the product. So if the assembly is too large and logistically becomes a nightmare to handle, you could potentially have lots of bioburden areas where it becomes difficult to offer sterility. So sterility needs to be designed into the initial assembly design when the process is being thought of, to get that bioburden level correct, to make sure the surface area isn't too large, and to ensure that from a vendor's point of view that sterility can come with the pre-packed ready to go manifold or assembly to the end user, to so that they can they don't have to worry about the sterility. To put it in a nutshell, that onus then becomes on the vendor, who become part of the end user's quality system. So there's a hidden layer of complexity that the end users of the assemblies don't actually see, which is the assessment of the sterility, making sure that everything that is delivered is up to the safe standard that it needs to be.
Yeah. Definitely, Graeme. So navigating the complex regulations is a challenge, and manufacturers need to stay ahead and informed of the regulations and adapt their processes accordingly. I think another challenge is also supply chain vulnerability. And I mean, what we have seen lately are a lot of dual sourcing initiatives, knowing that it can be challenging for custom assemblies and take effort upfront to prepare all of the necessary documents for regulatory agencies.
Lots to think about, and that's very good advice. Thank you. Angela, let me stick with you. Setting regulations and supply chain aside, what are some broader industry trends that you're seeing right now?
So, Graeme, you might agree on that. Some broader industry trends we are seeing is, first of all, increased demands for advanced therapies like cell and gene therapies, mRNA treatments that require flexible solutions that support fast changeovers. Then there's also a growing pipeline for monoclonal antibodies and biosimilars. So speed to market here is critical. And what we furthermore see is a shift towards decentralized manufacturing. So that means flexible modular units that enable local production to enhance supply chain resilience and reduce transportation costs. And also the adoption of digital and smart manufacturing. And another key trend is continuous manufacturing, which very well aligns with the just above mentioned technological advancements like automation and data analytics.
Thank you for that. You've both mentioned GMP and Annex 1. Graeme, I'll turn to you. Can you explain what GMP and Annex 1 are?
Yes, certainly. So GMP stands for Good Manufacturing Practice, and essentially it's about consistency and control through your process to make sure that that final product that you make is safe for its intended use. Annex 1 is a set of guidelines which is more around the contamination control within the industry. So it really looks at the overall process right down to the components coming into, at the start of the process to the vendors who are manufacturing the single-use assemblies to get through to the manufacturing facilities of the drug product, making sure that the bioburden levels, the particulate levels primarily are all controlled through that system, and that they have, and I'll use the word again, control and robustness in their processes for the ultimate goal of quality and safety.
And with that understandable emphasis on sterility, what specific challenges do you see companies facing now? What should they be thinking about?
I think I mentioned it before. The design of the single-use assembly is key to ensure that it can be manufactured with the low bioburden to be able to give that sterility. So the contamination control strategies that Annex 1 outlines and asks people to keep in place all the way down to the raw materials, the vendor of the single-use assemblies through to the manufacturing facilities. That is a key part of controlling all of that. So if you look historically, there was a very big reliance on sterile filtration to give that sterility at the end of the process. People would keep contamination low, but they would be reliant on that sterile filter, and that once they'd done their processing, they would test the filter to make sure that it was integral. It didn't have any issues with any particulates or any contaminants getting through. But on occasions, it had been found that there was what they call floor masking, where the process could be run, and then because there'd been lots of product going through that filter, any issues or irregularities in that filter could have been blocked by particulates or by some kind of contamination at the end of the process, which meant that anything which goes through the filter could have been compromised. So Annex 1 puts a bigger emphasis on all of this, and it's asking the end users or the manufacturers of the drug product to test the filters before and after use at their point of use. So the filters would go through a very long journey from the manufacturer where they would be tested for integrity, but then they're shipped around the world, they're incorporated into single-use assemblies prior to use. So, there's no guarantee the filter hasn't taken a knock and got some issues. So this Annex 1 regulation, asking people to test the filter before and after is now becoming key to ensure that the quality, the safety and the sterility of the product there. And it's a term that they've kind of nicely named PUPSIT.
And so you've obviously already led into it a bit, but any more you would tell us on what PUPSIT is and also the impact on organizations?
Yeah, certainly. So PUPSIT, it's an acronym which stands for Pre-Use Post-Sterilization Integrity Test. It does exactly what it says on the tin, basically. You're testing pre-use, you're testing it after it's been manufactured into a single-use assembly, so post-sterilization, and you're making sure that it's safe for product. So it's tested at that point of use, and primarily we're in a very risk-averse industry, so it's there to to make sure that safety, the quality, and I've mentioned that quite a lot during this discussion. Safety and quality are key, and they are behind everything which is done.
And Graeme, I think that's also where automation comes in, right? As automation is a compliance enabler here, a quality gate, and it simplifies and error-proofs the process. It reduces the risk of human error and ensures consistent processing.
Yeah, certainly it does. And, we have a variety of different automated systems, from standard filtration, tangential flow filtration, all the way through to bulk fill, and also including PupSit. So yes, it's all about control, it's all about error proofing, and it's all about consistency.
Outstanding. Thanks for filling us in on PupSit. As we wrap up, what are your thoughts now on the future of biopharma processing?
To me, it's consistency, control. So probably automation. That allows a computer is much better at controlling the accuracy of something down to the nth degree than a human can. So having that robust, repeatable control within your process is key. So yeah, I would say automation in the industry is a key thing to have.
Yes, totally agree, Graeme. And let's not forget about sustainability, as we often get questioned on that topic as well.
Yeah, plastics, right?
So the more and more companies are eager to have a beneficial environmental impact. However, studies and papers show that single-use technology, despite being disposable, can decrease the overall energy and water use compared to traditional setups. Another key for future will be collaboration and innovation. When vendors, manufacturers, and regulatory work together, this can only enhance safety, speed, and quality.
Yeah, I guess we're all one world. We all need to work together.
A great message to end on. Graeme, Angela, thank you both so much for sharing your insights today. Thanks to you.
Thank you, Mark.
It's clear that biopharma processing isn't just about technology. It's about delivering better outcomes for patients and enabling progress across the industry. To our viewers and listeners, thank you for joining us. Stay curious, stay informed, and keep an eye on how innovations are helping shape a better tomorrow. Bye-bye.